Hello, everyone.
Hi.
So, this is Jessica Sage and Barbara Hirst from RSSL. We are food safety and quality consultants who specialise in the area of allergen management. Normally, we would be out and about at the moment in factories and visiting our clients at their sites to advise on allergen management related issues. Clearly, we can't be doing that at the moment, given the current situation. So in light of that, we thought, why not have just a chat between us and share some thoughts on social media with the rest of you. And we thought what we would do was to start with think about allergen cleaning validation as a topic. Because, even though we are in the situation we're in at the moment, cleaning validation is still something that has to be done and has to be done well. And it's still very important, obviously from a safety perspective. So yes, we thought, why not have a chat about it and share our thoughts with the wider audience. Idea. So I guess on that note then, so why do we need to do allergen cleaning validation?
I guess very simply, really, if you've got a factory or manufacturing lines where you've got different products with different allergens in, and cleaning is your method to break between them, so you've got non-dedicated production, you've got to show that you can remove the allergen from the line before you make the next product that doesn't have it on a simple level. And you just need evidence, really, don't you, Jessica,
yep.
to really show that you've done that effectively.
Absolutely. And yeah, that is so much of what this is about collecting evidence. We do a lot of workshops and training around risk assessment as well. And again, as part of that, we focus a lot on the collection of evidence. It's all about having the evidence to know that what you're doing is sufficient and is good enough and is doing what you need it to do.
Absolutely.
So on that note, so moving on to thinking about actually planning the cleaning validation study itself, we see quite a lot of variability in how well cleaning validation studies are planned out. So Barbara, what would you say are your top tips for what a good plan looks like?
Yeah, I think it's really important to have thought about it in advance, to work out how you're going to actually conduct the cleaning validation, what's going to be included, what isn't. Make sure you record it, you write it down, and any decisions that you make, record that rationale so that, when that person who's actually done it maybe leaves or isn't there again, then you want to be able to have the evidence. So when you think about planning, you don't have to validate every line in every factory for every allergen. You can pick a representative line if they're cleaned in the same way, and you can pick something called a target allergen, so you don't have to do all your cleaning validation for all the allergens you might have on site. So that's really the important part of a plan.
And I think we do often see cases, don't we, where a manufacturer might be trying to do a cleaning validation study on every line that they have for all of the allergens that they handle. And really that's going to be really expensive and really time consuming for them. So if you can, pick a target and pick one line to use.
Yeah, and that's a really important part. This is an expensive exercise, so you really should be, when you're collecting evidence, collect good evidence, but you only need to take as few a number of samples as possible to answer that question of, was the cleaning good enough? And you mentioned target there, Jess. I guess we can talk about that a little bit. So if you've got lots of different allergens in your factory, you're making lots of different products, you're talking about how to show that the cleaning is good enough. So when you pick a target allergen, you can pick a product that contains a lot of a particular allergen, high percentage in the recipe, something that ideally has got quite a lot of protein in, because it's the protein people react to, and something that is particularly difficult to clean, because you can then show, if I've managed to clean that one, then everything else also would have been cleaned. You also want to pick a target allergen based on the fact that the laboratory that you're going to use has got a good validated ELISA testing method. And this is something I know in the lab in Reading, they do every day. They're forever, having to help people decide from a range of products and ingredients which ones should they pick. So really important to have thought about that in advance.
Yeah. And we would absolutely say, where you can, make sure you're using ELISA testing, really, because you want that quantitative test. And obviously, as you said, it's looking for protein, which is more clinically relevant in that respect, because it's what people react to, but also it's likely to give you a quantitative result.
Yes, absolutely. That's important.
What other? So we've thought about choosing a target. So the types of sample that you're likely to need to include in your validation study, you're going to need to do your clean samples. You're going to need a positive control sample as well.
Yeah, and that's something that people often forget about, I think, that they haven't done. So the best practice really is to take a sample of the actual product that contains that target allergen, so whether it's finished product or ingredient, whichever, send that to the lab to make sure they can actually detect enough of that protein in that product and that there aren't weird things going on really with the testing. Yep. Then clearly you do the clean, and you want to be doing a clean, following a standard procedure, good practice and all the rest of it. And then you'll take samples from all the different surfaces. There'll probably be swabs, they might be rinse waters if you have a CIP system. And then you really want to take the next product that's made that didn't contain the target allergen. And that's a really important thing because that's what the consumer's going to be exposed to. So that's a really important thing not to miss out. And the lab might need to do some validation and such like on it. But so that's kind of what the plan, I guess, looks like.
Yep, absolutely. I guess, so thinking about the next offline product, positive controls, I think from our experience sometimes those are the bits that might get forgotten, almost like the top and tail of the study. And as you said, positive control, that's really the only way you're going to know that what you're choosing as your target is something that's there in a high level, because clearly what you're trying to demonstrate is that your clean removes the allergen from the line and it removes it from a high level down to a non-detectable level. Yep. When we're thinking about results then, so we've done our plan, we've taken our good samples. What results come back? So clearly you're aiming for non-detectable results for all of your samples. And ideally, best practice is that you get 3 rounds that are completely clear. But often what we'll see...
That's what you aim for.
...is sometimes you might get positive swab results, but your next offline product comes back as negative. So, and there's often quite a lot of debate, and we often get asked, is that good enough? So, Barbara, over to you on that one.
And that's a really good thing. Is it good enough? That's really what you were asking at the beginning. Is my cleaning good enough? So, what do results look like? And again, part of your plan in advance should be to think about what am I going to do when I get the results back. So from your swabs or rinse waters or whatever it is, you want them all really to be to a less than that limit of quantification with your ELISA test. But if some of the swabs come back, maybe with a little bit has been detected, or even maybe a lot, and a lot would be a greater than, you want to be looking and thinking, well, where were those positive samples? Where did they come from? So was it a piece of equipment that was very difficult to clean? Or maybe the cleaning wasn't done so well at the time it was actually done. Was there a reason for that positive? But don't panic at this early stage. You need to investigate. Clearly, the next offline product you want not to be able to detect allergen in at all, because that, as we say, is what the consumer is going to eat. So that's the most important sample in this. But you want to get to the point where all of your swab results and all your rinse waters and the next offline product are not detected. And you need to do this on a 3 times over, 3 consecutive rounds to show repeatability. Basically is what you're really looking for. But if you get a odd positive swab result, don't panic, do a proper investigation, then repeat.
Yeah, absolutely. And we've emphasised so much the importance of planning and trying to not do this in a hurry, but we obviously also understand that at the moment time is quite a precious commodity. People are quite time scarce at the moment given the current situation. So, if you need advice on putting together a plan, what a good plan might look like, we can help and provide advice around that. But we can't under or over emphasise the importance of a good plan. Is that fair to say, Barbara?
Absolutely. That is absolutely fair to say. And I guess just one other last thing to mention is something people fall into a trap of is sometimes you see swab results on the plan or on the record reported as parts per million or percentage or something, and really all you can detect on the end of the swab is what's on the end of the swab. So it should be reported as an amount per swab. And don't fall into the trap of trying to translate that into what might be in the next product. So don't change it into a parts per million or a milligram per kilogram. It really should just be an amount per swab. It's just an indirect measure of how good the cleaning was. No more, no less.
Absolutely. Okay, brilliant. So I think we've covered most of the things we wanted to in this brief session. Unless did you have anything else you wanted to add, Barbara?
No, not really. I just, we're here to help, and we can do it remotely. So yeah, give us a call.
Wonderful. Well, we hope you found this useful, and who knows, this could be the first of many. We'll see about that.
Take care and stay safe, everyone.
Take care.